Hello, my name is Stewart Fisher, and I'd like to welcome you to the Mastering Medicinal Chemistry Conference, which will be held May 21st and 22nd at the West End Waterfront Hotel in Boston this spring. This exciting conference will cover a number of hot topics of interest to medicinal chemists, including allosteric inhibition, natural product chemistries, covalent inhibition strategies, and the role of binding kinetics in drug discovery. But first, allow me to say a few words about the current environment facing medicinal chemists today. It is clear to me that the field of medicinal chemistry is undergoing fundamental changes that range from the increasing demands to deliver innovative molecules against novel targets and mechanisms that were once seen as difficult or impossible, to confronting the challenges and opportunities of a globalized outsourcing marketplace. Importantly, the first session of this conference addresses these aspects with 4 talks from leading scientists from the major pharmaceutical firms. Hans-Joachim Bohm of Roche, Mark Bunnage from Pfizer, Mike Hahn from GlaxoSmithKline, and Jörg Hollenz from AstraZeneca. These talks will certainly provide important insight and potential opportunities facing medicinal chemists in their current roles, but I also believe they will provide a framework to better understand how medicinal chemistry is likely to evolve in the future. There are several topics that I find particularly appealing in the conference schedule. First, I think that strategies that utilize allosteric binding pockets for enzyme inhibition and receptor binding are a particularly hot topic at the moment. While past drug discovery efforts have focused on conserved active site architectures for lead identification and optimization, there are now a number of precedents where biomolecular interactions once thought to be undruggable, such as protein-protein interactions and large macromolecular assemblies, are now amenable to small molecule inhibition through allosteric mechanisms to achieve remarkable selectivity and potency. I'm really looking forward to the talk by Dr. Lindsley, who is scheduled to talk about the optimization of phospholipase D inhibitors that utilize an allosteric mechanism to achieve specific inhibition of this important enzyme, since I think this will both describe the unique features of allosteric site binding and the strategies required to exploit these opportunities. Second, I think the historical role that natural products have played in drug discovery is indisputable. But the industry has moved away from this source of leads for some time now due to difficulties in isolating new compounds and optimizing the leads following identification from extracts. I think that the talk from Dr. Peter Hamley from Sanofi-Aventis will be enlightening, as he is scheduled to talk about his company's approach and future opportunities in this area. Third, the increased utilization of covalent modification as a strategy for improving potency and durability of treatment is particularly intriguing. Historically, these approaches have been viewed as too risky due to the potential for off-target modifications. But Dr. Thijs Rivong from Pfizer will be giving a talk on using this approach against Bruton's kinase for the treatment of autoimmune diseases. I think this work will provide a useful case study for delineating the challenges and advantages of these approaches.